So our second talk is by Sarah Kearsley. She's a master's student at Western University. She can be found on Twitter at Kearsley Sarah. And her talk is titled Bilateral Stimulus-Locked Responses in Upper Forelimb Muscles During Visually Guided Reaches. So thank you, Sarah, and we're really looking forward to this talk.
There we go. Okay, so am I good to go now?
Yep, you're good to go.
Okay, awesome. Thank you. Good morning or afternoon, depending on where you are. Thank you for coming to my talk. I'm Sarah. I'm a first-year master's student, and today I'm going to be presenting the work from my undergraduate thesis project about rapid visuomotor responses in the upper limb muscles during a bilateral choice reaching task. We make visually guided reaches every day, such as when reaching to pick up a TV remote. It takes time to integrate the information about the visual stimulus, such as its size and location, to initiate a contextually appropriate motor response. It takes approximately 250 to 400 milliseconds to initiate one of these responses, which is fast, but I'm interested in situations where we might not have that much time. In these situations, like the one the picture was just in, we can get visual information to the muscle very quickly, at times that are approaching the minimal afferent and efferent conduction delays. I'm interested in what pathways underlie these rapid visual motor transformations. This question is complex given the pathways involved in visual and motor control. So here we're looking at some of the pathways involved in processing the visual image, reaching control, and eye movement control. Interestingly, there's this common node, the superior colliculus, in both of these pathways, and I'll actually be telling you how it's implicated in reaching too. The superior colliculus is a phylogenetically old midbrain structure involved in orienting movements and rapid eye movements, otherwise known as saccades. It's been studied extensively in regards to saccades, and I'm going to talk a bit more about its activity because it becomes relevant for reaching. So if we record from the intermediate layers of the superior colliculus during a task where a saccade is required to a newly appearing visual stimulus, we see three distinct types of neurons. There are neurons which show a purely visual response, time-locked to the onset of the visual target. There are neurons which show a motor burst, that is neural activity associated with the saccadic eye movement. And then there are these visual motor neurons. These neurons show both the visual burst and the motor burst of neural activity. If we look at the activity of these neurons across many trials, we can clearly see the time-locked burst of the visual activity occurring around 100 milliseconds, followed by the saccade-related bursts. As the saccadic reaction times become shorter, these bursts become closer together. This gives rise to bimodal peaking, the first peak associated with the visual response, and second with the motor. When these peaks merge into one, we get what are called express saccades. Express saccades are visually guided saccades occurring around 100 milliseconds towards the visual target. The superior colliculus's ability to mediate these rapid visually guided saccades make us think it may be involved in other rapid visual motor responses. The superior colliculus sends both the visual and motor information down to areas in the reticular formation for eye movements. More than just this, it sends to areas involved in head movements and arm movements. And from here, the information goes bilaterally. In line with this, if we stimulate the colliculus, we actually get contralateral and ipsilateral arm movements. But what happens to the visual information at these other premotor centers? This brings me to the stimulus-locked response. If we record from the pectoralis major, a muscle involved in cross-body reaches, we can look at how the muscle is responding across time during reaches to a newly appearing visual stimulus. Lower muscle activity is indicated by blue, with higher indicated by yellow. And we can see that the first wave of muscle recruitment actually occurs around 100 milliseconds. This is the stimulus-locked response. If we look across many reaches to a newly appearing visual stimulus, we can see that although the reach reaction time varies, shown by the black dots, this first wave of muscle recruitment consistently occurs around 100 milliseconds. Properties of the stimulus-locked response have been shown through other studies to actually be very similar to properties of the visual response within the superior colliculus. So there's an accumulation of evidence suggesting that this response is mediated by the superior colliculus via the tecto-reticulo-spinal tract. For my project, I wanted to further investigate the actual involvement of the reticulospinal tract. Stimulation studies and anatomical studies have shown that a key feature of the reticulospinal tract is that it projects bilaterally to the upper limb muscles. So I predicted that in a task where either arm could reach to intercept the visual target, the response would appear bilaterally regardless of which arm was chosen. To test this, we collected data from 15 healthy participants. Participants were seated in the KinArm endpoint robot, holding onto 2 robotic manipulandums. The task was displayed on a screen horizontal to their chest level, with their precise hand locations displayed on the screen. Throughout the reaching task, we recorded muscle activity bilaterally from the pectoralis major, the muscle involved in the crossbody reaches. Participants completed a bilateral hand choice emerging target paradigm. The emerging target paradigm has previously been used in a unilateral reaching task to reliably elicit the stimulus-locked response. We modified this paradigm to allow for reaches from either hand. Participants were instructed to initiate each trial by moving their hands into the white dots on the screen and looking at the notch in the barrier. Once their hands were in the dots, a target would appear at the top and move down to behind the barrier. It then re-emerged below at one of 7 possible locations. Participants were instructed to wait until it re-emerged and then reach as fast as they could with either hand to intercept the target. We collected 200 reaches to each target location, resulting in 1,400 reaches for each participant. Here, we're looking at the percent of right hand reaches as a function of target location. Not surprisingly, for the targets furthest to the right, the participant always reached with their right hand. And for the targets furthest to the left, they always reached with their left hand. But for targets that were closer to center, there was actually a mix of which hand reached. We were able to use the proportion of right arm reaches to make a psychometric function and find the point where 50% of reaches would be made by each hand. From here, we found the actual location with approximately an even number of reaches. We did this for each participant. This location was really interesting to us because we could look at what was happening in the right pectoralis major when the right arm was chosen to reach, but also what was happening in that same muscle when the left arm was chosen to reach to the exact same visual target. We could also look between muscles what was happening with it throughout a given trial. So here we see that when the right arm performed the reach, and if we're looking at the right pectoralis major, although the reach reaction time varied, we still get this initial wave of muscle recruitment occurring around 100 milliseconds. So we see that this version of the emerging target paradigm still does evoke the stimulus-locked response in the reaching arm. But more than just that, we could look on these exact same trials what was happening in the left pectoralis major. And what we found was that we still get this initial wave of muscle recruitment occurring around 100 milliseconds. Thus, we see the stimulus-locked response in both the left and right pectoralis major, even though it was the right arm that was chosen to reach. Further, we could look at what was happening within a muscle based on which arm was reaching. So if we're looking at average muscle activity throughout time, we can see that for the left pectoralis major, when the left arm reached, we get this initial burst of muscle activity, followed by sustained increased activity. Further, when the right arm reached within this exact same muscle, we see a very similar initial burst, followed by minimal muscle activity. The same pattern is seen in the right pectoralis major, with the sustained activity for the right arm reaching. Thus, we can clearly see the stimulus-locked response appears bilaterally, regardless of which arm is reaching. We were further able to look at qualities of this response, such as its magnitude and latency, based on whether the arm was chosen or not chosen to reach. And so what we found was that the response was actually about 20% larger when the arm was chosen to reach. Here we're showing the within muscle values for when that arm was chosen to reach and not chosen. Using a paired t-test, we also found that the response occurred about 4 milliseconds earlier when the arm was chosen. But this is a bit more inconsistent across participants. So we found that the stimulus-locked response does present bilaterally regardless of arm choice, supporting reticulospinal tract involvement in the response. Further, on the chosen arm, the response was a bit larger and tended to occur a bit earlier. Most importantly, this first wave of muscle recruitment during these rapid reaches is bilateral during the stimulus-locked response interval, but then rapidly gives way to lateralized recruitment favoring the chosen arm. I think it'd be interesting in the future to look at the pathways underlying this initial burst of muscle activity in animal models and further in clinical populations. I would like to thank the various members of the Corneal Lab and the various funding sources for this project. Thank you.
All right, thank you, Sarah. That was a great talk and super, super clear. So I'm really, yeah, I think it's great work. I had a few questions. We also have plenty of time if anyone else has questions. I'll defer to the audience first. Joran Smeets has said the effect of the chosen arm must be based on a prior. For instance, the choice in the previous trial. Did you check for that? So how much did one trial influence the subsequent trial?
I haven't looked at that yet, actually, but I did want to look at that because I do think like if you reach with the right arm 3 times in a row, that likely does influence the next one. But because for this outside trials, you always get the reaches with the closest hand, I do think that they are taking in the information from the trials, but that'll be interesting to look at in the future.
Great. Yeah, that's an interesting point. So I am going to ask my question. If you can go back to the time course of activation, muscle activation for the 2 arms, what it looked like to me was that there, actually you can see it in both. There's the peak at the stimulus-locked response, and then there's a subsequent peak in that about 150 to 175 millisecond range.
Yeah.
Both in the arm that's reaching, more so in the left, there's kind of 3 peaks, but in the right, there's just like the second peak and rise. But there's also in the arm that's not reaching, you see 2 peaks. I was wondering if you could talk a little bit about what that might mean to you.
Yeah, so this actually follows with previous work that was done looking at the stimulus-locked response. So there's a paper by Wood et al. 2015, and they found that there's an oscillatory behavior that's at about 15 hertz that actually falls in line with what you'd see from the reticulospinal tract. So we think that this has to do with that oscillatory activity. And yeah, so
just to follow up on that, is it then that when the reach is being made, is it necessary for both pectoralis muscles to contract to stabilize the trunk to make the reach? And that's why, like, at the initial point of the reach, you would have these, or is that not the case, that actually this is a product of the colliculus being only delivering bilateral responses, so it not being hemi-specific? Does that make sense?
Yeah, so I don't think this has to do with postural stability, because if you look, the response is like very large, like it's half of the size to almost the size of the actual voluntary activity. So I think that it's just basically like, because the colliculus does orienting and stuff like that, that you're just like, it's the first initial wave of preparatory activity for it, and then after that it gives way to the lateralized reach.
Great. So that's really, yeah, I think it's a great answer. We've got another question in the Q&A from Ivan, who says, "Hi, Sarah, super clear talk. Congrats. I was wondering whether the effect was due to some kind of motor overflow or because of a stabilization strategy caused by a slight torso rotation." So this is similar to my question. But also adds this concept of muscle overflow. Do you have any ideas about that?
I'm honestly not sure. I'm not sure about that one. Sorry, I'll have to think more about that.
That's no worries. And you can always pick it up in the chat with Ivan if you guys want to discuss further, because I think it's an interesting point. So I think that